All right. As Peter said, I'm Arjun Prasad and on the pathogen detection team at NCBI, and I'm going to tell you a little bit about how to get access to our data in Google Cloud and how to work with it a little bit. But before I get there, I wanted to tell you a little bit about where the data comes from and how it's generated. So we have whole genome shotgun sequence of isolates that are submitted by collaborators at the FDA, CDC, USDA, state and local public health labs, international public health labs, basic researchers, clinical sources, et cetera. They submit their whole genome shotgun sequence data to the SRA, and then when we know about it, we pick it up in our pipeline. So what is that pipeline trying to do? That has two primary goals: to answer the question, are these isolates clonally related? Could they be part of the same point source outbreak or things like that? And then the other is to identify the antimicrobial resistance genes and point mutations to assist with AMR surveillance and research. And we present the results of this pipeline in an easy-to-use web interface, and importantly for this talk, on Google Cloud. So how do we do that? A very brief overview of the pipeline is that we take the whole genome shotgun sequence data from the SRA and we assemble it with our in-house developed assemblers that I'll tell you a little bit about in a moment. And then we cluster those assemblies by comparing them to all the other assemblies for the same taxonomic group in our system, as well as any other assemblies we can find for the same taxonomic group in GenBank. And then we cluster them into these groups of very closely related organisms, and within each of those clusters, we infer phylogenetic trees. At the same time we're doing that, we annotate and run AMRFinderPlus, and I'll tell you a little bit more about AMRFinderPlus as well, and that is how we identify antimicrobial resistance genes, point mutations, virulence, and stress resistance genes. So first, about our assemblers. We use two assemblers, Schiza and Sauté. The first, Schiza, is a high accuracy, high speed de novo assembler. And this was developed to make the clustering less likely to have errors because it makes very few errors in the assembly. Most other assemblers are really trying to increase your contig length, and so they can make sometimes leaps that are a little too far in joining contigs. Whereas Schiza, it basically just assembles along until it comes to a part that's difficult to assemble, where it thinks it might make a mistake, and then it just stops. So that means that it's the contigs can be a little bit shorter. And to increase sensitivity, we also use the Sauté guided assembler. So this is a high sensitivity assembler that can pull apart alleles based on variants represented in at least two reads. So if you had a well-known carbapenemase gene like KPC2, and we have isolates that have like a KPC2 and KPC3. Both of these can only be one nucleotide apart, and Sauté is able to pull apart those multiple copies. And if that organism had two different genes that were only one nucleotide apart, we get the two different genes. The guides for the Sauté assembler are made from AMR genes and STX. So what this means is that our assemblies are made up of the Schiza de novo assemblies with the Sauté guided assemblies added on. And so that means that our assemblies in GenBank will often include duplicated contigs. And you could tell them the contigs are usually duplicated where there's an AMR gene that was assembled by the Sauté guided assembler. And you could tell those contigs apart because the ones assembled by Schiza will have de novo in the name, and the ones assembled by Sauté will have guided in the name. And we'll see an example of that in a little bit. Now, to tell you a little bit more about AMRFinderPlus, it identifies acquired AMR genes, known AMR-associated point mutations, plus some intrinsic AMR genes and relevant stress and virulence genes for some taxa. And what makes AMRFinderPlus different from a lot of other antimicrobial resistance gene finding software? Well, there's a number of things, but I think the biggest thing is that it makes an affirmative statement as to what the gene should be called. So AMRFinderPlus is really trying to give the sequence the name that one of our curators would give it if they were looking at it manually. So if you had a KPC2, as I mentioned, which is a named beta-lactamase allele, only sequences that are 100% identical at the amino acid level would be named KPC2. If there's any variation at all, we'll tell you that it's either another named allele or that it's an unnamed allele, and we'll just call it block KPC. So how does this, how do we present these results? Well, in Google Cloud, we have two primary outputs from this analysis pipeline. We have the isolates browser, where every row is an isolate genome, and columns are metadata provided by submitters or calculated based on the genome assembly. And I'm going to show you these very briefly in our web interface to give you some orientation. And we also have MicroBigQuery, the microbial browser for the identification of genetic and genomic elements. And here, every row is a gene or point mutation that was identified by AMRFinderPlus. Columns are metadata about the isolate or gene or point mutation or how it was found, or the reference sequences in our database. And this contains results from public assemblies in GenBank. So when we assemble things, they may first appear in the isolates browser, and then we submit to GenBank, and there's some delays and sometimes some QC issues that mean that not everything makes it into MicroBigQuery. So there will be a slightly smaller number of isolates there. So I'm going to switch over to web browser and show you real quick our web interface. This is the pathogen detection homepage, and I'm going to go over here to the isolates browser. And I'm going to type in a search for Inoki. I'm just picking this because there were a couple of recent outbreaks of listeria that was from contaminated Inoki mushrooms. And so here you can see this is the main table view. And here every row is an isolate assembly, and every column is information about that assembly. So here, isolation source, you can see fresh Inoki mushrooms. You could see this was isolated in Taiwan. These are the clusters of closely related organisms. We can click into those and you can see the trees. And you could see here there's an environmental isolate and a clinical isolate. They're closely related. So you can see how this can be used to investigate outbreaks. But I'm not going to tell you too much more about that. The other thing I wanted to show you is MicroBigQuery. So I'm going to go into MicroBigQuery and look at the genes and point mutations identified by AMRFinderPlus in those isolates. So here you can see this is MicroBigQuery. Every row is a gene or point mutation. You can zoom in a little bit. And every column is information about that, either information about the isolate, the element that was found. So these are the gene symbols here, or how the isolate was found by AMRFinderPlus. All right, so that's all I was going to show you in our web interface. Now let's get to the cloud. So we are using Google BigQuery to present these tables in the cloud. And what is it? It's a managed SQL database on Google Cloud. It is designed to easily and quickly allow you to search large public databases. So why are we using it? Well, we have a large database we want to make public. And in this way, we can give you bulk access to all 21 million rows of MicroBigQuery, including the updates that we do every day. And it also uses standard SQL query syntax, so you can do complex queries and analysis just in Google BigQuery without having to download anything. Google has a quite extensive documentation on BigQuery. I have some links here. And we have a number of example queries in our documentation. And if you are trying to figure out how to use this and you've looked at these resources and still can't figure out how to get the information you want, you can feel free to email us at pd-help@ncbi.nlm.nih.gov. So I don't have time to teach you all about SQL. There's a lot of good resources on the web to learn SQL, but in case it's new to you, I wanted to just give you a few little lampposts about how things work. So SQL queries are kind of separated into separate sections, and one of those is the select clause, which tells you which fields you want to get from the tables or from the query. The from statement says where you want to get the data from. And importantly, because SQL is, or BigQuery is a relational database, that means that you can link multiple tables on shared fields. So you can join, and we'll see an example of that in a minute. The where clause tells you which results you want. Give me the results that meet these constraints. We'll also use group by, which is how you cluster results. We use it for counting. You can use it for statistics, things like that. So this just clusters all the groups of rows into a group. And order by to sort the results. There are a number of SQL operators. Some of the ones you may be familiar with are the normal ones: greater than, equal to, less than. They work as you would expect. There's also the like operator, which allows you to search with wildcards. So this, in this case, the percent is kind of the equivalent of a star in Linux or DOS. There's also an unnest. We'll see an unnest function, which is used to flatten a repeated field. So even if you're familiar with SQL, you may not have come across unnest before. We'll see in a minute, but Google BigQuery allows you to have fields that can contain multiple values. And those values can even have multiple subfields within them. And we'll see this, but unnest allows you to just flatten them out and treat them like a table. So let's go to the Google Cloud web interface. Here is when you get to Google Cloud, if you type up BigQuery in the first cloud interface, you can click over here and you'll get to the BigQuery web interface. Now, the first thing I'm going to do is add a data source. I'm going to star a project, and all of our data that we're going to present is in this project, NCBI Pathogen Detect. And so if we star this, then it appears over here, and we can use that to kind of browse it and see some more information about it. So this is the information is in the PD browser database, and these are the tables that we have there. Here you can see the isolates table and the microbiggy table. There's also this isolate exceptions, which I'm not going to tell you much about, except that that's for QC check failures and reports those results. But if you click on any of these tables, you can see their structure. Here's the schema, which is just the fields that are available there. And here, this AST phenotypes field is one of those complex fields I was mentioning, where it's a record field, which means it has multiple kind of subfields, and it's repeated. So there can be multiple entries, and we'll see what that looks like. You can also click on microbiggy and see things there. There's a tab up here with more details about any of the tables. So here you can see there's 21 million rows in microbiggy right now. And you can preview and see what the data actually looks like. So here you can see some of the entries in the table. Okay, so let us start running some queries. So kind of to start simple, here's a query just to say, give me some information about a specific isolate, if I had an isolate I was interested in. So we're going to select, and these are the fields we want to get back from. And then in BigQuery, tables they're indicated by putting the table name with the full paths of the table in backticks. So that's the key above the tab on US keyboards. And so NCBI Pathogen Detect is the project, as I already mentioned. PD browser is the database, and isolates is the table. So we want the rows in there where biosample accession is equal to this. And all you have to do to get the results is click run, and you get those results. And so you can see here, this is one of those repeated fields. There's multiple values for this row in that field. Okay, let's do something else. So this is very similar to what we did before in the browser. We looked for Inoki, but here we're going to count the results. So this is just the way you count things in SQL, and we're going to get the from the isolates table, where the isolation source is like %Inoki %. So that just means 0 more characters of any kind, followed by Inoki, followed by 0 or more characters of any kind. And so when you do this, this will identify any isolates or count any isolates where Inoki appears anywhere in the isolation source field. So we click run, and we get that there's 246. Now, if you were really eagle-eyed, you might have noticed this number doesn't exactly match what I showed you before, and that's because these searches are case sensitive. So if there's a capital E in Inoki. So if we just shortened it like this and click run again, you'll see we get a larger number. So there were some times when the person who submitted the isolate capitalized the word Inoki. So let's look at something a little more complicated. So say we want to find contigs that have a lot of AMR genes. So antimicrobial resistance genes often come on contigs or in plasmids that are transferred between bacteria, and they can be really nasty. So let's just say we wanted to identify the ones that were kind of the worst, the really bad ones. So here we're going to identify the contigs with the most AMR genes. So we're selecting from the contig accession, and we're using that count, and we're going to rename the count as the number of genes. So that's the number of rows in microbiggy that returned from this query. And then we're also going to count the number of unique gene symbols. So we're using this distinct function, and we're going to call that number of unique genes from microbiggy, where subtype equals AMR, so that's just AMR genes. And we're going to use the group by clause. So we're going to get one row for every contig accession. And then we're going to sort the results by the number of genes. So we defined num genes up here, and then descending. So we get the biggest numbers first. And we're just going to take the top 10. So if we click run, you can see there's these contigs with lots of AMR genes. And this first one, this looks kind of crazy, right? There's 96 AMR genes on one contig. but it looks a little fishy to me because there's only 16 unique gene symbols. So that means some of them have to have been repeated many times, and that seems a little odd. So let's take a look and see what else is on the contigs that we found in this query. So here's the query we just ran, and you could run it again, and we're going to use this in what's called a subquery. So we put that query in parentheses. And here we're going to select all the rows, this information from MicroBiggie. We're renaming MicroBiggie or aliasing MicroBiggie to MB. And then we're going to join that. Remember how I said you can join between tables? We're joining to the results of another query. And that other query is the one we just ran, and we're going to call that top 10. And we're joining on where the contig accessions match. So we're going to get all of the entries in MicroBiggie where that contig appears in this query we just ran. And then, of course, we're going to sort by the number of genes, which is what we did before, as well as the position on the contig, just so the genes kind of come in the order they do on the contig. When we click run, we get the results. So here's this contig, and you can see it has, let's make a little more space here. You can see it has that 96 genes, but only 16 unique genes. And if we look down at what those genes are, you can see, oh, there's this big run of APH3'-1As. This doesn't seem really biologically plausible. I think there may have been an error in the assembler got stuck in a loop or something here. So as always, you have to be careful about the data and look at it. And I will say this isn't one of our assemblies. Okay, so let's look at one of those complex fields. So here, we're going to look at that AST phenotypes field that I talked about. It's a repeated record field, and we're selecting from the isolates table. And here we're using another subquery, but this time it's in the where clause. And here what we're going to do is we're going to use this unnest function to kind of unroll that repeated field, and we're going to count the number of entries where the antibiotic is %penem. So we're looking for carbapenem resistant things that tested with AST data to be resistant to carbapenems. Now, carbapenem antibiotics all end in the suffix penem, so like ertapenem, meropenem, doropenem, et cetera. And so that makes it easy to write this query, right? And then we want the ones that are resistant. And so when we count those, we want where there's one or more carbapenem resistant AST tests for that isolate. So we click run, and you can see the results here. And so there are about 1,500 isolates that are were tested carbapenem resistant to one or more carbapenems. Just to get really fancy, although I'm not going to run through it in detail, this is a big query to identify all the carbapenem resistant isolates that we saw before, where no mechanism for carbapenem resistance was identified by AMRFinderPlus. So these are carbapenem resistant isolates where we couldn't tell what the source of the resistance was. So this would be a good place to look for novel mechanisms of resistance. And if you click run, you get those results, and you can see these are isolates that are resistant to one or more carbapenems. Okay, and then the last thing I wanted to do was to show you how to get access to the sequence data. And so what we're going to do is we're going to use the contig URL. There's a contig URL and protein URL entry in every row in MicroBiggie, and that is kind of an address of where to get the sequence data in Google Cloud. So these are in Google Cloud Storage buckets, and here is the address of the data. And I'm going to just use the Cloud Shell, which is did I copy that? I think I did not. Which is a way to get a Linux prompt. Here it is. And then we're going to use gsutil. So you could do this from your own Linux computer, as long as you had the Google Cloud Shell utilities installed. And we're going to copy that URL to the local directory like that. And it's going to ask me to authorize the Cloud Shell to access Google Cloud for it. And there we go. There's the file we just downloaded. So that's the sequence of the contig. We can unzip that file and and look at it. And so here's the sequence of the contig. And this looks like this is a de novo contig, so it came from Skiza. This is not one of the guided contigs, and you can see here's the sequence. And so you can do the same thing for the protein sequences as well. So that's about all I have time for right now. And I think I can go for questions.
Thanks, Arjun. So there's a few questions in the questions pod, a couple of general questions. First, what would you say the advantages are of using the cloud versus trying to do these kinds of queries some other way? Is the cloud the only way you can do them?
So a lot of the things I showed you can do using our web interface, but some of the more complex analyses you can't do with the simple search syntax that's available on the website. So that would be one of the things. The other thing is the micro-biggie data, the 21 million rows that are updated daily. We don't have another way to present that data in bulk other than the web interface. So if you wanted to do some kind of bulk analysis that you couldn't do with the web interface, this would be the only way you could do that. The other thing is that this gives you easy access to all of the contigs. So all of these assemblies that I'm presenting or showing in micro-biggie are available in GenBank, but this is just kind of a more convenient way to get easy access to specifically the contigs and protein sequences you might be interested in.
Okay, great. So these whole genome shotgun assemblies that you've made, those are available potentially from our FTP site as well as they are
Yes,
from the
absolutely,
cloud.
Absolutely.
Okay. There was another question that always comes up for these it's about cost. Mm-hmm. And Bill and Michael Felgarden answered a little bit in the Q&A, but can you comment on how much? So, for example, the queries that you ran today.
Sure. So when you write a query in Google Cloud, it shows you how much data it will process, and Google charges based on the amount of data you process. The thing is that the amount of data we have is very small relative to their costs, and they provide 1 terabyte of free search per month. And so it ends up being that I never go over a terabyte, in using this stuff. So it ends up always within the free tier. But even if I were to have to pay for it, it would be, these will be a few cents at most, these queries.
I'm going to answer the viral one. So there is a whole other work being done, especially on COVID, but I don't know exactly the extent of all the viral sequence databases. That's a completely different group at NCBI. Maybe Peter can comment on that.
Yeah, so the question is there an equivalent process for something like NCBI viral? So certainly for the sequences, there is, especially for COVID and the SARS-CoV-2 sequences. So we have a website called NCBI virus, and that would be the way to access the viral data at NCBI. Another question that's in here that hasn't been answered yet, is there an API for the SQL? I'm not sure I completely understand that question.
So there's certainly, there is an API for Google BigQuery in several languages, including the command line as well. And you can find information about that in Google's BigQuery documentation.
Okay, and I think somebody asked about so you did show you have a web page where people can go to get the queries that you're using at BigQuery, right?
Yes, I think I linked to it here. Here we are. So I put them up here, but we also in our documentation have a number of other queries. So I encourage you to check out our main Google Cloud documentation. There's a lot of many additional queries there that show you different things you can do. And of course, as I said, if you still can't figure things out, you feel free to email us.
Anyway, we'll share that on the FTP site that's available.
Yes.
I'll link to the slides that are in the chat as well.
All right. There's some other. I have the Q&A open now. So the number of protein genes. So we don't have the counted number of protein genes in each of the assemblies in our tables. You can go to GenBank to get that information for any of the assemblies, and that's available also on the FTP site for the total number of protein genes. How much difference does it make when we use assembly to identify AMR genes rather than finding AMR by short reads only? So it depends on your methods. AMRFinderPlus is specifically designed to work on assembled sequence, and so we can take advantage of some things like an actually annotated sequence. We can take advantage of protein HMMs to be able to better identify and classify novel genes. We also because you have the assembled sequence, we can say this is the allele represented in the assembly. And so we're very, AMR Finder Plus, as I mentioned, is kind of focused on getting the name right. And so we can do that from the assembly. It's not designed to run on reads. I guess that's what I would say the advantages are.
Right, just sort of a general question, Arjun, and maybe Bill would want to answer this. So one of the main points of the pathogen detection project is to help to make these associations between illness outbreaks and the source. Do we have any estimate for how many outbreaks have been solved by this process?
Well, FDA's said on their website that they've used the pathogen detection system since 2013 for over 1,000 public health actions. Those don't always mean outbreaks, but it is indicative of both the frequency of foodborne illnesses and how often pathogen detection has been used. And on our homepage, we have a little panel over on the side called Success Stories, and that lists some interesting publications, government websites that sort of show or highlight very successful uses of pathogen detection for various facts, identification of novel genes, solving outbreaks, etc.
Very nice. Yeah, so Arjun's showing that now in the slide. Let's see if there's any more questions. I think we've answered the ones that are open. Okay, well, thanks everybody. And I will put these questions together in the ones that have been answered and put them in a document and make them available on that FTP site link. So thanks everybody for coming. And thank you, Arjun, and thanks Bill and Mike for answering questions.
Thank you.